Alright, so for the third lecture on Chapter 10, we're going to look at how the muscle contracts. Recall all the material from the first two lectures on microscopic muscle anatomy and on the excitability of our skeletal muscle cells, what action potentials are, what resting membrane potentials are, how they're maintained, because you'll need all that knowledge to understand how skeletal muscles contract. So there are three main phases to skeletal muscle contraction. First, we have the excitation phase. With the excitation phase, this is where an action potential arrives from the nervous system at the neuromuscular junction and sends a signal to the muscle fiber by releasing acetylcholine into the synaptic cleft. It binds to a ligand-gated channel. Remember back our ligand-gated channels. And when that ligand-gated channel opens, our sodium floods in, causes depolarization. And once we have this local, so this is a local depolarization because of the ligand-gated channels, once we have this local depolarization, then it opens the adjacent voltage-regulated channels. And so this is our excitation-contraction coupling. So we're going to couple our original excitation to our actual contraction down in the muscle fiber. And so we couple that by sending that action potential via voltage-gated channels. So we're depolarizing all along here, depolarize, 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 all the way down into those T-tubules. Remember, our T-tubules are the invaginations of our sarcolemma. So this is an invagination of the actual plasma membrane, the sarcolemma of our muscle fiber. When that enters down into there, we enter the triad. Remember, adjacent to the T-tubule are our terminal cisternae, which are part of the sarcoplasmic reticulum. It stores calcium. And so the sarcoplasmic reticulum releases its calcium stores. That calcium comes down here and binds to the troponin. And remember, this is one of our regulatory proteins. So like I said, you need to refer back and remember our microscopic muscle anatomy. And so this starts the contraction phase. So three main phases: excitation, excitation-contraction coupling, and then the contraction phase. And so now that we have the calcium bound to the troponin, it changes conformation, pulls the tropomyosin off of the actin active sites. Once the tropomyosin is moved off of the actin active sites, now the myosin head can bind. This is called cross bridge. So the cross bridge is formed. The myosin heads ratchet forward, pulling the actin towards that M line, shortening our sarcomere and causing a muscle contraction. All right, so let's look in more detail at our neuromuscular junction. So we have the axon coming in. It terminates in a terminal, so we have our axon terminal. The axon terminal is filled with synaptic vesicles containing the molecule acetylcholine. Embedded in the axon terminal are these voltage-gated calcium channels. The axon terminal comes into close proximity but never touches the actual sarcolemma of our muscle fiber. And so the gap that is in between there is called our synaptic cleft. The area in which we have the synaptic cleft is referred to as our motor end plate of the muscle fiber. And within that motor end plate of the muscle fiber, we have several ligand-gated channels that are specific for acetylcholine. They have the acetylcholine receptor on them. And so that's the makeup of our neuromuscular junction. And so again, our excitation phase. So excitation phase is our first phase of muscle contraction. First step of excitation is an action potential arrives at the axon terminal. When that action potential arrives, and remember this is depolarization, repolarization, so we are changing the polarity of the axon's membrane. and that causes voltage-regulated calcium channels to open. Calcium flows in via passive diffusion. When that calcium flows in, it triggers these synaptic vesicles, which contain acetylcholine, to fuse with the axon terminal membrane, release the acetylcholine via exocytosis. Once the acetylcholine is released from the axon terminal, it diffuses across the synaptic cleft, binds to the ligand-gated channels, and because of our sodium-potassium pump and our leak channels, well, mainly our sodium-potassium pump, we have a higher concentration of sodium outside the muscle fiber than inside, and so when that acetylcholine binds to that ligand-gated channel, opens the channel, the sodium flows in. When the sodium flows in, recall back to our action potential that the motor end plate is depolarized, and that's why it gives it this electrical yellow glow, is because we have a local, so this is a local depolarization producing what we refer to as an end plate potential. So these are the 6 steps for excitation in our neuromuscular junction. So this is step 1 of muscle contraction, the excitation phase, which consists of 6 steps for it to occur.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so that video is basically what we talked about, but it had a few extra steps stick with the 6 steps that I give you here. And the end of that video where it was talking about the removal of acetylcholine by acetylcholine esterase will come up when we talk about the relaxation phase of the neuromuscular junction. All right, so let's watch. That was an AP flicks. And so under the study area, you can find the AP flicks there. And this next one, we're going to look at the big picture animation. And so it relates more directly to the image that we're looking at now.
Let's look at the structure of the neuromuscular junction before we examine how it works. The neuromuscular junction includes the enlarged distal ends of the axon, called axon terminals, and the junctional folds of the muscle fiber. The junctional folds are the region of the muscle fiber membrane, or sarcolemma, with specialized structures for receiving signals from a motor neuron. Let's look more closely at a single axon terminal and the adjacent junctional folds. Notice that the axon terminal and the junctional folds do not touch. They are separated by a gap called the synaptic cleft. The membrane of the axon terminal contains voltage-gated calcium channels. These channels will open in response to an action potential, allowing calcium ions to diffuse into the cell. Within the axon terminal are membrane-bound synaptic vesicles that contain the neurotransmitter acetylcholine. Calcium ions will cause these vesicles to release acetylcholine into the synaptic cleft through exocytosis. On the other side of the synaptic cleft, the junctional folds contain acetylcholine receptors. The acetylcholine receptors are chemically gated ion channels that open when they bind to their ligand, acetylcholine. Outside the junctional folds, the sarcolemma contains voltage-gated sodium channels. Once the muscle fiber is stimulated, these channels open, initiating an action potential on the sarcolemma. This action potential will trigger muscle contraction.
Skeletal muscle fibers contract only if stimulated by a motor neuron. The connection or synapse between a neuron and a muscle fiber where this stimulation occurs is called the neuromuscular junction, or NMJ. The neuromuscular junction forms between the axon terminal of a neuron, which is the expanded end of a long process called the axon, and a portion of the muscle plasma membrane called the motor end plate. Note that no physical contact takes place between the neuron and the muscle fiber. Instead, there is a narrow space between the two called the synaptic cleft. We will now look at the steps of the first phase of muscle contraction, called the excitation phase, in which a neuron stimulates or excites the muscle to contract. First, an action potential, or electrical stimulus, arrives at the axon terminal. The action potential then stimulates the synaptic vesicles to release a chemical called acetylcholine into the synaptic cleft. Acetylcholine is a chemical known as a neurotransmitter because it transmits the stimulation across the synapse. During the third step of excitation, acetylcholine quickly diffuses across the cleft and binds to its receptors on the motor end plate. Acetylcholine receptors are ligand-gated cation channels that open and allow sodium ions to pass when acetylcholine binds to them. Notice the folds in the motor end plate. These increase its surface area, so there could be many acetylcholine receptors concentrated close to the site of acetylcholine release. As positively charged sodium ions enter the muscle fiber, a local depolarization develops in the motor end plate. We call this depolarization an end plate potential. In the next section, we will look at how this electrical change in the muscle fiber spreads and leads to contraction.
Okay, so those last 3 videos summarized or went back over our excitation phase. Remember, the excitation phase is the first phase in muscle contraction. And so in the next video, we will look at excitation-contraction coupling along with actual contraction. And if you want to view those 3 videos, they are in the study area. The links in the PDF that I posted may work, I'm not sure. But you can find these as the AP flicks and as the big picture animation in the study area of mastery. Have a good day and let me know if you have any questions.